Good morning. This is Michael Dong speaking on an introduction to HPLC. Here is the agenda. Two questions we'd like to answer. First, what is HPLC? Here we describe what is HPLC, a brief history, and the separation mechanism. Second question is why is HPLC so useful? Here we explain the benefit of HPLC and showcase a example in pharmaceutical analysis. What is liquid chromatography, LC, and HPLC? Liquid chromatography is a physical separation technique where a sample is separated into its component by the distribution between a mobile phase and a stationary phase. HPLC started in 1960s when liquid chromatography became high performance or high pressure using a high pressure pump to force mobile phase through small particle columns. UHPLC stands for Ultra High Pressure LC, which is a modern HPLC equipment platform with high operating pressure of over 15,000 psi. And also the equipment has lower dispersion in the system. Here's a brief history of HPLC, starting with 1906. A Russian botanist called Mikhail Tvek used this terminology, chromatography, to describe color writing. In 1941, two biochemists, H.A.P. Martin and R.L.M. Sink, published on partitioning chromatography and became Nobel laureate in 1952. In 1966, the first HPLC equipment was developed by Professor Chapel Horvath of Yale University. In 1969, a small company called Waters Associates introduced the first commercial HPLC called ALC-100, and later on introduced one of the most important pumps called M6000, pump capable of 6,000 psi operation. In the 1970s and 1980s, HPLC became almost indispensable for routine use for industries and research. And these are due to advances in bonded phase chemistry and the use of small particle column and the development of UV detector, which is sensitive and reliable. This slide describes a brief history of HPLC from 1990s to present. In 1999, Professor James Jorgensen of University of North Carolina and Chapel Hill published a proof of concept research on UHPLC. Followed 7 years later, in 2004, Waters Corporation introduced the first commercial UHPLC system rated at 15,000 psi. It was called the Acuity UPLC. Later on in 2010 and later, LC mass spec emerged as a powerful tool for scientific discovery and research. Two important references for your reading. One is an article in C Engineering News published in 2016 called 50-Year HPLC. Second one is a book chapter called UHPLC in Pharmaceutical Analysis in the book called Chromatography, A Science of Discovery, published in 2010. This slide describes traditional low pressure LC, which is a predecessor of HPLC. In this case, happened quite often in an organic chemistry laboratory for purification, where a silica gel column was fed under gravity with solvent for purification of organic mixture synthesized in the laboratory. Fractions are collected, and analyzed manually by UV or by other techniques such as mass spectrometry. This slide shows a schematic diagram of an HPL system, including the four major module: pump, injector, detector, and CDS, stand for chromatography data system. The flow path starts with the mobile phase in solvent reservoirs, shown here, followed by degassing to get rid of the oxygen and nitrogen in the solvents, and then go into the pump, which is a high pressure pump, which is precise, which fed the solvent into an injector and or auto sampler, allowing the introduction of the sample. The sample would flow into the column inside a column oven, and then into a detector, typically a UV ultraviolet detector, before the eluent was wasted. All the modules are controlled by a CDS, a chromatography data system, which also happen to do the data handling. In this slide, we illustrate four different HPLC equipment from the four major manufacturers of HPLC, which control about 85% of the market today. Start off with Waters Acuity UPLC, which is an automated liquid chromatography, or UHPLC, with a very precise pump, very precise auto sampler, and an online detector, which is UV, plus a CDS, which is a PC or a client server. Next is the Agilent Technology 1260 UHPLC, followed by Shimatsu iProminence HPLC, and finally the Waters Scientific's Vanquish UHPLC. This slide describes the chromatographic process in three separate diagrams. First one is the overall separation, showing a column, which is a column packed with solid support or silica, with the sample being a mixture of two components, A and B. And while the mobile phase is flowing through the column, component B is being retained by the column and then eluted later, while component A, which is less retained, emerged first. Now, in diagram C, the chromatogram, which is a recording of the detector output, in this case the absorbance versus time, you can see that the B component emerged later as a peak, which is broader, while A eluded earlier in this case. B is an interesting diagram which showed the microscopic view of the chromatographic process. Here we have a solid support, typically a porous silicon material. The support is bonded with a ligand. In this case, most likely it's going to be a hydrophobic type of ligand. And we have two analytes, B and A. You can see that A is less retained from the partition coefficient of the arrows, which is going less into the stationary phase, while B is emerging later because it's highly retained by the hydrophobic stationary phase. Here we want to answer why is HPLC so useful. So we summarize the benefits of HPLC. Also, we like to summarize what are the perceived limitations. So the benefit of HPLC is, number one, is wide applicability, which is applicable or amenable to very diverse analyte types, starting from the pharmaceutical, proteins, ions, polymer, to pesticide residues. Number two is precision and reproducibility for quantitative analysis. Number three is when HPLC is coupled to a mass spectrometry, which has become the most powerful tool for scientific discovery. And finally, HPLC have high separation power, coupled with very sensitive detectors such as UV detector and mass spectrometry. However, there are some perceived limitations which happen to be mitigated or reduced by recent development. First limitation is no universal detector. Nevertheless, a UV detector and a detector called CAD or charge aerosol detector is getting close to be a universal detector. The second one is lower separation power or efficiency when compared to capillary GC. Nevertheless, the emergence of UHPLC, this perceived limitation is kind of getting away because UHPLC can deliver almost the same efficiency as a capillary GC. Not quite as much, but almost. Finally, HPLC is arduous for regulated testing or QC testing, which is still true today. Here is a case study in pharmaceutical development. We'd like to illustrate some of the major application of HPLC. The sample is a drug product, which is a tablet extract. And this assay is called a stability indicating UHPLC assay, meaning that it is a purity assay where the main component is being measured, plus all the impurity and degradation product in the drug substance or in the drug product. And this is a drug product. The top chromatogram is a standard mixture. This happened also to be a retention marker standard, kind of a calibration standard, where the main component called the API or active pharmaceutical ingredients is spiked with some of the expected impurity or degradation product. The molecule of the API, the structure is shown here in the inset. In the lower chromatogram is the sample. The sample happened to be a drug product extract, which is a tablet, which has been subjected to an accelerated stability condition at 50 degree and 75% relative humidity for 3 months. The column and operating condition are shown here in the inset with the full information shown here. And as you can see, one can see the main component and some of the expected impurity peak being shown in the samples readily. Finally, here is our summary and conclusion. To answer the question, what is HPLC? HPLC is the common analytical technique using the liquid as the mobile phase and the column packed with stationary phase or bonded phase operating at high pressure. Its main use actually in quantification of various types of sample. Why is HPLC so useful and so common as an analytical technique? The main reason is really HPLC can deliver very precise and reproducible analysis of a wide variety of sample type from pharmaceuticals to protein peptides and small ions. Here are 2 references that can provide more information. First one is this book published in 2010 by Lloyd Snyder, Jack Kirkland, and John Dolan called Introduction to Modern HPLC. The second one is a book published in 2019 called HPLC and UHPLC for Practicing Scientists, published in 2019. And the material came from chapter 1 of this book. For more information, the audience is directed to some of the websites, nwd-consulting.com and also chromeclass.com. Also, you are urged to subscribe to the channel for later episodes. On this note, I'd like to thank you for your attention, and we see you next time.